Unit 2, we're going to talk about structure and functions of cells. So in this activity, we're going to study cell types and microscopes. So station 1 you did at your desk, so you should have already read pages 6 through 10 and answered the questions in your packet. Once you turn in that packet, you're then going to get another packet, and at that point you're going to start the presentation, which you are doing currently. Remember, you can pause, and you should pause at I would say the stop signs, but in this program there aren't stop signs. Just you're going to have to make sure that you push the pause button. So station 2, we're going to start looking at the history of the microscope. People in ancient times knew that a drop of water that fell into a grain of sand or small insect made the object or insect it fell on look larger than it actually was. So it was Seneca at 4 BC who then lived till 65 AD. He was a Roman philosopher who would enlarge letters by filling a glass globe with water. So a little trivia here. So what Pixar movie showed this process of magnifying objects using a drop of water? Now, Ptolemy was a Greek astronomer who lived in the 2nd century. He was the first person to explain why this happened, why water magnified objects. So he understood how bending light could magnify these objects. Ptolemy was also the first astronomer to suggest that the Earth was not in the center of our solar system and that the sun was. So here you can see a picture of that heliocentric theory versus geocentric theory. So of course we understand that the sun is in the middle of our solar system, but for the longest time it was believed that the Earth was in the middle of our solar system and everything revolved around it. So when we look at microscopes, they were first used during the early 17th century. We don't know who invented it because several different types were used during this time period. Many discoveries were made by Robert Hooke and Van Leeuwenhoek. Now Hooke looked at cork cells. He is who named the cell the cell. So when we look at cork cells, they're dead cells, so they're hollow, they don't have much inside of them. And when he looked at them, it reminded Robert Hooke of the rooms that monks stayed in, which were called cells. This is why he named them cells. Now, Anton van Leeuwenhoek, he is known for his advances in the microscope. He viewed many living microscopic organisms, and the video I'm going to show you next is going to show you with Bill Nye the Science Guy and him using a Van Leeuwenhoek microscope. So now looking at the compound light microscope, which is what we'll be using in class, it bends light through an object and we can magnify the image. Very important that you pay attention to that detail, that light has to be able to pass through the specimen or else we cannot magnify it using this tool. You will need to get yourself familiar with the parts of the microscope so when we use them, if I ask you to put something on the stage or to use the fine focus, you know what I'm talking about. Now, station 3 is going to get you a little familiar with the 3 types of cells. So the first type of cell are referred to as prokaryotic. These are bacterial cells which do not have any internal organelles except for ribosomes. So you can see we have the outer covering here, which is the cell wall and the cell membrane, and then we can see some other structures which include the DNA. So the DNA is right in through here. So you can see the DNA in this nucleoid region. It doesn't have a nucleus, but the DNA sits in, kind of compacts itself in one area inside there. The only organelle that we will see inside a bacterial cell is the ribosomes. So here are some bacterial cells. So we have our streptococcus right here, which streptococcus is something, if you guys get strep throat, that's what that bacterial cell looks like. Of course, these have been stained so that we can see them much easier under the microscope. The other bacterial cells over here are bacillus. They're more of a rod-shaped structure to their cells. So the circles we call coccus and the rods we call bacillus. The next type of cell we'll look at are plant cells. So if you notice, they're rectangular in shape. They're more boxy. They have a thick cell wall. So when we're looking at that line right there between all of the cells, those are cell walls. So these are all cell walls. And the green dots inside them, all right, we can see the green dots. Those are called chloroplasts. And the last type of cell we'll look at are animal cells. Now these particular cells here are cheek cells. Notice that the animal cells they're more round in shape. They have a very clearly stained nucleus. They do not have a cell wall. And then you might be able to see these little stained dots on the surface of the cells. Lots of them here. Those are bacterial cells. So once you get to this point, you have one more packet to pick up, and that packet is going to actually have you getting your own microscope and looking at a couple specimens. You're going to look at the letter E, and you're going to make some observations as to how it behaves under the microscope. And we're also going to look at some thread. You're going to take different colors of thread, and you're going to cross them underneath the microscope slide that you're going to make. And you're going to notice how there's not only a field of view. The field of view is the area you see under the ocular, but there's also a depth component. And so there's different layers that you can zoom in or focus on. So we're going to look at that in this next lab packet that you're going to get.